Yo, welcome to the deep dive. I'm Arav and Priya ma'am, I need to file a complaint against NCERT.
Oh boy, ये तो interesting होगा. What happened?
मैम मैंने DW का 4 hour lecture देखा on ATP synthesis. Then I checked NCERT page 161. And both are teaching that one glucose gives 38 ATP. But then I Google modern biochemistry research and it says 30 to 32 ATP.
Finally, someone actually fact-checked NCERT. अरव, ये 38 ATP वाली वैल्यू 1960s की है, जब तुम्हारे दादा दादी स्कूल में थे तब की. Modern mitochondrial research clearly shows realistic yield is 30 to 32 ATP.
But ma'am, ये तो official textbook है, CBSE approved. How can they be wrong?
Let me tell you a corporate horror story. Last month, a global hotel chain sent an email to a male principal addressing him as Dear Miss Pandey. Corporate incompetence, no fact checking, copy paste from outdated databases. NCERT is doing the exact same thing. They are copy pasting 1960s ATP values without updating for modern P/O ratios, proton leakage, or transport costs.
Miss Pandey? That's actually tragic. But okay, so NEET में क्या लिखना है? 38 या 30?
The million-rupee question. देखो, NEET technically follows NCERT. तो अगर question says, according to NCERT, total ATP, write 38. But अगर they give you modern P/O ratios, 2.5 ATP per NADH, 1.5 per FADH2, and ask you to calculate, answer is 30 to 32. You need to know both systems.
So basically, NCERT ने हमें एक outdated map दिया है. But we need to know the real terrain भी.
Perfect analogy. And DW, Done Academy? बच्चों, 38 ATP याद कर लो. No explanation of why it's outdated, no modern science, just blind rote. तो आज, we're doing a full audit. The logic behind both calculations. Ready to infiltrate the truth?
Born ready. Let's expose this ATP scam. Okay, so पहले basics. Where does chemiosmotic theory actually work?
Inner mitochondrial membrane. Think of it as a high security casino vault, Arav. Electrons are the currency coming in, protons are the chips being moved around, and ATP synthase is the cashier converting everything into biological money, ATP.
Wait. So what exactly is chemiosmotic theory?
Brilliant question. Peter Mitchell proposed this in 1961, won a Nobel Prize in 1978. The theory says ATP is not made directly by electron transport. Instead, electron flow creates a proton gradient across a membrane, and this gradient drives ATP synthesis. It's indirect, two-step process.
So electrons don't directly make ATP?
No, this was revolutionary, Arav. Before Mitchell, everyone thought electron transport directly phosphorylates ADP. But Mitchell said, nahi bhai, electrons pump protons first, gradient banta hai, then ATP synthase uses that pressure to make ATP. It's like a hydroelectric dam. Water doesn't directly make electricity. It spins a turbine which makes electricity.
Oh, so the proton gradient is the pressure.
Exactly. And here's where it happens. Inner mitochondrial membrane has the electron transport chain, ETC, complexes 1, 2, 3, 4, and ATP synthase, complex 5. As electrons move through complexes 1, 3, and 4, protons are actively pumped from the mitochondrial matrix into the intermembrane space.
So the intermembrane space becomes crowded with protons.
Yes, high concentration outside, low inside. This creates an electrochemical gradient, both a concentration difference and a charge difference. Mitochondrial matrix becomes negative, intermembrane space becomes positive. This is the proton motive force.
And ATP synthase uses this force.
Exactly. ATP synthase has two parts, F0 embedded in the membrane, F1 sticking out into the matrix. Protons flow back through F0 And this flow makes F0 rotate, which forces F1 to synthesize ATP. Physical rotation creates chemical bonds. Your mitochondria is literally running a nano turbine.
That's actually insane. तो ये कॉन्सेप्ट फोटोसिंथेसिस में भी होता है?
Yes, in chloroplast, thylakoid membrane में, same principle. Proton gradient drives ATP synthesis, universal mechanism. But NCERT barely connects the two. They teach them as separate chapters.
Alright, walk me through the ETC. How do electrons actually move?
Okay, so imagine a highway with four toll booths. NADH from glycolysis and Krebs arrives at complex 1, NADH dehydrogenase. Electrons are stripped, NADH becomes NAD+, and as electrons pass through complex 1, four protons are pumped out into the intermembrane space. First toll booth. They transfer
Got
to
it.
ubiquinol,
Where do electrons
also
go next?
called coenzyme Q. This is a mobile lipid-soluble carrier that shuttles between complexes. Now here's the twist. FADH2 from Krebs cycle enters at complex 2, succinate dehydrogenase, feeds electrons directly to ubiquinone. But complex 2 does not pump protons.
Wait, so FADH2 skips the first toll booth?
Exactly. This is why one FADH2 produces less ATP than one NADH. Fewer protons pumped means less gradient means less ATP. NCERT doesn't explain this. DW doesn't explain this. They just say FADH2 kam ATP deta hai. Why? Logic nahi dete.
Okay. So complex 1 pumps 4 protons. Complex 2 pumps 0. Next?
Ubiquinone carries electrons to complex 3, cytochrome BC1 complex. Another 4 protons pumped here. Then, electrons move to cytochrome C, a small mobile protein on the outer surface of the inner membrane. Think of it like a taxi between complex 3 and 4.
And complex 4?
Complex 4, cytochrome C oxidase. Final destination. Electrons combine with oxygen and protons to form water. This is where O2 is actually consumed. 2 more protons pumped. So total per NADH, approximately 10 protons pumped across the membrane.
And if oxygen is blocked?
Everything stops. Cyanide poisoning blocks complex 4. Electrons pile up, NADH can't be oxidized, Krebs halts, cell dies. Brain and heart affected first because they need the most ATP. Death in minutes.
Brutal. But clear. So now, how many ATP?
Here's where NCERT betrays you. ATP synthase needs roughly 4 protons flowing through it to make 1 ATP. So, 10 protons divided by 4 equals 2.5 ATP per NADH. But NCERT rounds this to 3 for simplicity. How simple?
And FADH2?
FADH2 enters at complex 2, skips complex 1, so only 6 protons pumped. 6 divided by 4 equals 1.5 ATP. NCERT says 2, again outdated.
Okay, the moment of truth. Ma'am, break down the full calculation. NCERT versus reality.
Alright, from one glucose molecule, here's what we get. Glycolysis, cytoplasm, 2 ATP, substrate level phosphorylation, 2 NADH, cytoplasmic, pyruvate to acetyl CoA, mitochondrial matrix, 2 NADH, 1 per pyruvate, 2 pyruvates total, Krebs cycle, matrix, 2 GTP equals 2 ATP, substrate level, 6 NADH to FADH2 total from 1 glucose, 4 ATP, substrate level, 10 NADH total, 2 FADH2. Now the calculations diverge.
Give me NCERT first.
NCERT, 1960s outdated model. 10 NADH times 3 ATP equals 30 ATP. 2 FADH2 times 2 ATP equals 4 ATP. 4 ATP, substrate level. Total equals 38 ATP.
And modern biochemistry?
Modern, realistic P/O ratios. 10 NADH times 2.5 ATP equals 25 ATP. 2 FADH 2 times 1.5 ATP equals 3 ATP. 4 ATP, substrate level, total equals 32 ATP. But there's a catch. The 2 NADH from glycolysis are made in the cytoplasm. They need to enter mitochondria. This costs energy.
Wait, it costs ATP to transport NADH?
There are two shuttle systems. Malate aspartate shuttle in heart, liver costs nothing extra. Still gives 2.5 ATP per cytoplasmic NADH. But glycerol 3 phosphate shuttle in muscle, brain feeds electrons to FADH2. So those two NADH only give 1.5 ATP each instead of 2.5.
So, depending on the tissue, total ATP can vary?
Exactly. In heart/liver, 32 ATP. In muscle/brain, 30 ATP. So the real answer is 30 to 32 ATP, not 38. But NCERT still teaching 38 because they haven't updated since 1985.
This is actually a scam. So, for NEET,
Read the question carefully. If it says, according to NCERT, write 38. If it gives you P/O ratios, 2.5 and 1.5, and says calculate, write 30 to 32. If it says theoretical maximum, 38. If it says actual yield, 30 to 32.
alright, give me the high yield NEET traps for chemiosmotic theory.
5 Goldmine questions. 1. Who proposed chemiosmotic hypothesis? Answer: Peter Mitchell, 1961, Nobel Prize 1978. 2. Where does oxidative phosphorylation occur? Answer: inner mitochondrial membrane, not matrix. Matrix is where Krebs happens.
Got it. Next,
What is the role of oxygen in ETC? Answer: final electron acceptor, combines with electrons and H plus to form water. Not provides energy. That's wrong. Oxygen doesn't provide energy, it accepts electrons. 4. Which complex does not pump protons? Answer: complex 2, succinate dehydrogenase. This is why FADH2 gives less ATP.
and number 5.
What drives ATP synthesis in oxidative phosphorylation? Answer: proton motive force slash electrochemical gradient slash chemiosmotic potential. Not electron flow directly. The gradient is the driver.
Mnemonic for the complexes?
NADH slowly cues, cytochrome accepts. NADH dehydrogenase, succinate dehydrogenase, Q ubiquinone, cytochrome BC1, ATP synthase accepts protons.
Love it. Clinical connection?
If ETC is poisoned, cyanide blocks complex 4, rotenone blocks complex 1, oligomycin blocks ATP synthase, ATP production crashes. Tissues with highest energy demand die first: brain, heart, kidneys. NEET loves asking, which poison affects
And the
which
difference between
complex?
chemiosmotic
Notice.
theory in mitochondria versus chloroplasts?
Same principle, different location. Mitochondria, inner membrane, protons pumped from matrix to intermembrane space. Chloroplast, thylakoid membrane, protons pumped from stroma into thylakoid lumen. Both use ATP synthase. Both are driven by proton gradients. Universal mechanism.
Alright, students, this was the deep dive, Summit Neuro. But before we go, there's something critical we need to tell you.
देखो, Summit Neuro is not a mass market program. We're partnering with only 5 more schools this academic year. That's it. After that, waitlist mode.
And we've already confirmed with rhymes with PPS and other rhymes with M, Amity International. Your rival school? They signed the MOU yesterday.
So, if you want Summit Neuro's logic first, NCERT audited, modern science integrated modules in your school, you need to create demand. We've made a Summit demand form, link in description. Download it, print it, get 50 signatures from your batchmates.
Then walk into your principal's office, not like Miss Pandey's email sender who doesn't fact check. I'm glad your husband, who's a solicitor, is handling them in court now, but with confidence. Say, Sir slash ma'am, our competition has Summit Neuro. Why are we still stuck with 4-hour DW lectures that teach outdated ATP values?
Because here's the brutal truth: DW will sell you a 100-hour course with 38 ATP calculations. Dun Academy will make an animated superhero called Proton Man. Seep Ma'am will say, Goddess of Biology has spoken. 38 ATP is final. Great. Meanwhile, we'll teach you both systems in 10 minutes with logic.
So infiltrate your campus. Demand biology that respects your intelligence. Demand modern science. Demand Summit Neuro. And until the next dive, remember, NCERT says 38, reality says 30 to 32. No both, ace NEET.
See you in the next dive, legends. And don't be the person who emails a male principal as Miss Pandey. Fact check everything.
Even your textbooks, especially your textbooks.